Thanks for joining us for this symposium. I'm Bill Rapisi, President and CEO of the Lymphatic Education Research Network. LE&RN's mission is to fight lymphatic diseases through education, research, and advocacy. In order to win a fight, you first have to join it. So we ask, please become a supporting member of LE&RN at lymphaticnetwork.org. And we hope you enjoy today's symposium. Thank you.
Maybe one day lymphedema will really have a cure, and that is my dream, and I think we should all participate actively in trying to make that happen. I'm going to try to touch on some of the horizons that are now visible, some of the developments that have occurred, and it gives me a chance to talk to you a little bit about my own research, so that's a little self-serving, but I hope it's going to be interesting for you as well. So this is not true about lymphedema yet. It is not a hip disease, but we're hoping to make it that. And maybe some of the things I'll talk about during this section will take us a little bit in that direction. I don't know how many of you watch CNN or are aware of this really huge breaking story last spring, but CNN helped us to understand that we found a new organ in the body, and it could be the biggest one we have. I can't tell you how many phone calls about this when it happened. So I'm glad the rest of the world now knows that we have the interstitial space, but it's good for lymphedema because that's our playing field. And if people think that's a great big new organ and think it's exciting and worthy of CNN, then maybe it'll shine a little bit of light on what we do for lymphedema as well. This is a concept that I try to emphasize every time I talk to general audiences, which is that lymphedema is very important and intrinsic diseases of the lymphatics are very important. But I believe, as I showed at the very beginning of my time up here, that image of the brick wall with the mortar, the lymphatics play a central role in health and disease in general. And once we uncover all the mysteries of the lymphatics, we're going to be a lot smarter about controlling all sorts of disease. So I'm going to show you a laundry list of conditions in which either the lymphatics have been demonstrated to play a role in pathogenesis or in treatment, and the list grows really month by month. So clearly we understand the vasculature of cancer and of cancer metastasis is heavily dependent on lymphatics. Autoimmune disease, we know that they play a role in rheumatoid arthritis and many other connective tissue diseases. There is tremendous similarity in the histopathology of lymphedema and that of scleroderma or progressive systemic sclerosis, just to name a very few of these. We know they play a tremendous role in chronic infection and inflammation. They have a role both in organ transplantation rejection and therapeutically by manipulating the lymphatic component in assuring that organ transplantation is successful. We now understand that the lymphatics in the heart play a central role in the repair process of myocardial infarction. They play a role probably in atherogenesis as well, based on their contribution to the reverse transport of cholesterol from the vascular wall through HDL. And they also play a role not only in the edema of congestive heart failure, but in mechanisms related to the function of the heart itself. They play a role in obesity, metabolic syndrome, and diabetes. I think we've touched on some of that today, but the topic is actually much bigger than what we've been able to talk about. And most recently, because we have demonstrated now that indeed, in contradistinction to what was previously believed, there are lymphatics in the central nervous system. There is a growing belief that lymphatics play a very important role in the pathogenesis of degenerative diseases of the central nervous system, like Alzheimer's disease, multiple sclerosis, and others. So this is all the horizon for the future. I think lymphatic biology is going to play a tremendous role in the coming decade as we evolve into new therapeutics in general. I'm happy to say that we're making great inroads at the National Institutes of Health. I had the opportunity to work there for 4 years under an IPA agreement where I spent 1 week a month at the NIH, collaborating with the different institutes, trying to raise their interest and awareness in the problems. And this was an intramural conference that we organized, the first of the intramural conferences that we organized back when I was there as a direct employee. Since then, we've had 2 or 3 additional ones where I've helped to organize as an extramural liaison. We have a trans NIH coordinating committee that Bill Rapisi and I continue to serve on as we help to guide the NIH in how to better serve problems related to lymphedema and lymphatic disease. I'm happy to say that if there is a parent institute at the NIH, and it's the one I worked in, it's the National Heart, Lung, and Blood Institute, and they recently called for public input into setting their research priorities, and I'm happy to say that there are 132 research priorities that have been defined, and one of them is lymphatic. Now that's a big deal. 132 sounds like a large number, but when you think about the compelling issues that NHLBI deals with, we are on the table, and they're very interested in determining normal biological function and resilience of the lymphatic immune and non-immune circulatory function. So that's a real coup, I think, for our field. I'm happy to say that the field of published lymphatic research is growing at an exponential rate. If we were a board of directors of a company and this was our profits graph, we would be jumping up and down. So since 1950 or so, the growth of lymphatic-oriented publications has been truly phenomenal, and this continues right up until the present day. I'm happy to say and proud to say that I've served as editor-in-chief of Lymphatic Research and Biology since its inception 14 years ago, and I'm very happy to say that we just learned that for 2018 we experienced a 23% increase in our impact factor. So our queue of publications is growing quite dramatically. We've this year increased our publication frequency from 4 times a year to 6 times a year, and that's because the publications are happening. And as I go to national meetings and international meetings, I'm proud to see how often publications from this journal are cited in public presentations that scientists and clinicians are making. I'd like to move on now from sort of the horizon for future developments in lymphatics to talk about a couple of concrete issues. One is an area that we haven't been able to talk about much today, and that is lymphatic and complex malformations, but this has been a trying area for clinicians, much as lymphedema has been, in that any treatments that we've had historically have been initially surgical when they're feasible. More recently, we've been able to have some endovascular techniques, but they're really, non-surgical percutaneous techniques to create ablation of these anomalies. But when these anomalies are complex or when they involve microvascular abnormalities of the lymphatics, they're really not amenable to the percutaneous ablative techniques. But there is hope on the horizon, because over the last 5 to 8 years, it has become clear that we can manipulate lymphatic vascular growth through pharmacologic means. And by repurposing an older drug that is still currently used for organ transplantation and to suppress immune function, but this belongs to the broad category of agents called mTOR inhibitors, it's clear that a lot of these lymphatic malformations are driven by overactivity in this pathway, and that by giving a directive mTOR inhibitor, we can actually inhibit the overgrowth of lymphatic vasculature that leads to the clinical problem. So just to show you a couple of examples, this is a very profound head and neck lymphatic vascular malformation where the child was treated with sirolimus, and you can see, of course not resolution, but dramatic reduction in the size and the impact of this lymphatic malformation that otherwise would have no direct therapeutic intervention. So I'm going to switch now to some of my own research, which I'm very excited about. I hope it can excite you as well. And I've spent most of my career trying to edge toward the notion that we could introduce drug therapy to deal with lymphedema. Lymphedema falls into a category of medical problem that doesn't fit into the model of typical adult chronic diseases insofar as we don't have a pharmacotherapy for it. We treat it through physical means, we control it, but we can't send the patient to Walgreens or the local drugstore to pick up a prescription to try to deal with this problem. I've always believed that we could perhaps identify the substrate of the biology of the lymphedema and pacifies it. We probably aren't going to regrow lymphatic vessels anytime soon, but if we can help the patients to coexist with an impaired lymphatic circulation, we can render them less symptomatic and perhaps even return them to the stage 0 of their disease. That's really been the thought behind my work. And I'm not alone in focusing on the notion that inflammation is at the core of the pathogenesis of this problem. And what we've been able to find in our own research and some publication that is about to come out quite soon is that as we profile the plasma of these patients, in fact, there are pathways that are instigated by the presence of regional lymphedema that have to do with a lot of effector cell mechanisms in the skin and in the tissues themselves that actually drive this forward. So once again, I've shown you this progression from pitting to non-pitting. This has really been the core of the biology that I've been interested in. What drives this progression? Can we halt it? Can we reverse it? And so we have chosen to work in and establish an animal model that is useful for a rapid throughput and to allow us identification of appropriate approaches. So this is in the tail of the mouse. We can create lymphedema using the mouse tail as a surrogate for the swollen arm or leg. We devised mechanisms whereby we could actually measure the edema volume as much as we do it in the patient by using the truncated cone approximation, and we can study the molecular basis of the changes in the skin, which is where the action is. And basically what we found when we did this in early work back in 2006, again, this heavy component of inflammation that really intrigued us. It seemed to be most of the signaling that was happening at the tissue level, despite the fact that we were dealing with a vascular and edematous condition. And as I've shown you earlier, we then extended that work to human skin and found that very many of the same expression pathways are upregulated in human lymphedema as well. And 4 of these, as I've shown you earlier, actually have to do with the inflammatory and immune distortions that are occurring at the skin level. So again, it allowed us to elaborate this notion that chronic inflammation is really a driver in this disease, and if we could abrogate this inflammation in a very specific way, perhaps we could turn the clock back for these patients. Now, I say in a very specific way because we all know that there almost isn't a disease state described in medicine that doesn't have some component of inflammation associated with it. Inflammation is a protein manifestation of human biology in health and disease, and therefore anti-inflammatory therapy comes in very many flavors. And just saying, I'm going to treat this with an anti-inflammatory doesn't mean that you're going to be in the right vehicle any more than if you try to use your key to open somebody else's car. So we began our work moving forward from that work we did initially in the animal model and the pathways that we identified, and we determined that a very likely substrate that was active here was a pathway that doesn't get talked about quite so often in inflammation, which is the lipooxygenase pathway, or the leukotriene mediators of inflammation. And basically, we saw some signals in that work we did in 2006 that made us think that we wanted to impair the impact of this pathway that produces inflammation. And we alighted upon a drug that we were able to use in our studies that actually inhibited two pathways simultaneously. It inhibits what all the other over-the-counter NSAIDs do, which is the cyclooxygenase pathway. Any drug, Motrin, Aleve, Advil, that your patients can go into the pharmacy and buy over-the-counter will inhibit this pathway and inhibit the generation of thromboxane that is an inflammatory mediator. But the NSAID that we used, which is a prescription one called ketoprofen, in addition to inhibiting cyclooxygenase, also inhibits 5-lipoxygenase. And for that reason, we thought it was an excellent candidate to look at in lymphedema. And basically what we found is that when we treated these animals systemically for about 2 weeks with ketoprofen, thinking that perhaps combined inhibition of cyclooxygenase and lipoxygenase was the answer, we were quite delighted to see that not only did the edema go away, but the treated skin of these animals almost completely resembled normal skin, with almost complete resolution of all of the architectural changes that we see in untreated lymphedema. And I showed some of these images earlier today. And similarly, as I showed you this morning, where lymphedema causes these lymphatic channels in the skin to massively dilate, we found that after treatment for 2 weeks, these lymphatic vessels basically became non-visible again, much as they are in the normal skin. So we said, okay, this looks pretty real, and this is becoming important not only because it works, but because it addresses part of the dogma that may not be correct, which is that as we progress structurally in lymphedema, we're dealing with irreversible changes in the skin, because we were seeing the changes reverse, at least in the mice. So we decided to use this drug, which was able to be repurposed in humans, and conducted first an open label study and then a placebo-controlled study. And here you can see a typical subject in our study in the open label phase, where we decided to look at skin thickness with a caliper as a surrogate in the clinic for what we were seeing under the microscope. And basically, this is the same patient before and after 4 months of exposure to a systemic dosing of ketoprofen. The reduction in the skin thickness from the very enlarged pretreatment value to nearly normal at the end of 4 months. There was no other change in therapy. The patients who entered this study were required to have completed CDT, to be in a maintenance garment, to use the maintenance garments throughout the period of enrollment, and the only change in their treatment was the addition of the drug. As a point of reference, if I do this test on anybody in the room here who doesn't have lymphedema, I'll likely be able to subtend a piece of skin that's about 2 millimeters, maybe 3 millimeters in thickness. What you see on the left-hand panel is about 35 millimeters, and after treatment is down to about 3 or 4 millimeters, so nearly normal. So this is what we saw in the open label trial overall, and this, of course, achieves statistical significance. Not every patient was a responder, but the average response was quite a dramatic reduction. We also did the same thing in a placebo-controlled trial where the placebo controls did not respond, but the lymphedema patients did respond. We looked at 5-LO staining of specific subtypes in the skin to try to determine, indeed, was this in any way linked to the 5-LO mechanism? And again, we found that in lymphedema skin, the expression of 5-LO both in macrophages and neutrophils in the skin was significantly reduced by ketoprofen therapy, but not in the placebo-treated patients. I haven't shown you the histologic changes in either the open label or the placebo-controlled trial, but basically we devised an empiric histologic score based on the attributes that we determined on a training set of lymphedema specimens, and then we scored these, or the dermatopathologist scored all of our specimens in a blinded and random fashion, and we found that ketoprofen was associated with a dramatic reduction in the dermatopathology score, whereas placebo treatment did not have an effect. Well, we became curious at that point to say, all right, we're dealing with a drug that inhibits cyclooxygenase, it inhibits lipoxygenase. Can we give all our patients Advil? Is that really what's going on? Or is it something related to 5-LO. And for that reason, we began to look at a variety of agents back in the animal model to determine whether or which of these pathways was important. And basically, to make a long story short, we found that when you inhibit cyclooxygenase, a la Motrin, Advil, et cetera, you don't affect the lymphedema at all, or perhaps you make it worse. But when you treat with anything that inhibits in the 5-LO pathway, you make the lymphedema better. So that's what the upper panels represent. The lower panels I'll call the Advil or Motrin approach. So we looked at various components downstream of 5-LO, and we determined that if we inhibited below 5-LO or at 5-LO itself, we had a therapeutic benefit, but it was really by inhibiting products in the LTB4 pathway that we were able to achieve the therapeutic result. So basically 5-LO results in 3 pathways of downstream activation, and we learned that LTB4, leukotriene B4, was the modulator that seemed to be responsible for the lymphedema insofar as blocking its activation led to resolution of the lymphadematous changes. We looked at levels of LTB4 in human plasma as well as in the mouse sera, and we found that in both cases there were dramatic elevations in LTB4 levels in untreated lymphedema. So we became pretty convinced that if we extended this observation to the human condition, that we might have something interesting to look at. So again, looking at this broad context, we're eliminating cyclooxygenase as an area that we're interested in. In fact, it may have a detrimental effect to block cyclooxygenase because cyclooxygenase produces, among other things, prostaglandin E2, which stimulates lymphangiogenesis. So we really are interested in LTB4 specifically as an agent that is able to induce phagocyte attraction and activation and to reduce inflammation when it's when it is inhibited. LTB4 has a very complex biology, and I don't really have time to go into this today except to say there are lots of reasons why we would think that LTB4 would be an interesting molecule to block. When we used a specific LTB4 antagonist in the mouse model, we performed what amounts to a mouse lymphoscintigram. Here we're injecting a fluorescein-labeled dextran into the subcutaneous tissues. You can see what the normal pattern looks like in the tail with delineation of this hexagonal lymphatic capillary network under the scales within the mouse's tail. When the mouse develops lymphedema, it develops just what looks like one of those fluorescent images or like a lymphoscintigram with thermal backflow and sort of dissolution of all of the ordered flow that we would expect in the presence of a normal lymphatic vasculature. When we gave the LTB4 inhibitor, which in animal use is called bistatin, we almost completely restore what I'll call the mouse lymphoscintigram to normal within a period of 7 to 14 days. Pretty dramatic evidence that something interesting is going on by blocking this inflammatory inhibitor. I'm going to show you just one of these panels, if it works, which is to show you the equivalent of an ICG lymphographic study, but in the mouse tail, to show you that when we use the LTB4 inhibitor, we actually restore lymphatic flow proximal to the site of lymphatic obstruction. So if I can get my pointer there, this is where we do the surgery, this is where the lymphedema is, and there you can see the appearance of normal lymphatic flow proximal to the site of prior ligation. I'm not going to take the time to show you the saline treated animal, but I will tell you that in the absence of active treatment, you don't see that form of proximal flow restoration. So that's additional evidence that the drug is restoring not only lymphatic function, but its anatomic integrity. So to summarize LTB4 effects, when we overexpress 5-LO in vivo, there is an impairment in lymphatic drainage, excessive lymphatic sprouting, and microvascular leakage. When we antagonize LTB4, we reverse edema, improve lymphatic function, and restore lymphatic architecture in the mouse tail model. In vitro, LTB4 works at lower concentrations in a favorable way, whereas in the higher concentrations that we see in lymphedema, it inhibits lymphangiogenesis and induces apoptosis. We found that it's this pathway that is responsible for the impairment in lymphatic repair, and we have primarily identified the fact that it impairs signaling through VEGFR3 and through Notch pathways, both of which contribute to that repair mechanism. And we found that in part by creating Notch1 deleted mice, and they were not able to respond to the LTB4 because we removed that signaling pathway. I can just talk about this in a very prospective way, but we've just completed the first phase 2 trial of an LTB4 antagonist. The drug is called Ubenemex, and the acronym for the trial is ULTRA. We enrolled 55 patients in an international trial that just completed about 2 weeks ago. Dr. Stephen Dean had one of the sites and enrolled some patients there. We had patients enrolled in Sydney, Australia, and close to 40 at Stanford. This was a leg-only study of primary and secondary lymphedema. We are currently analyzing the results, and hopefully that's going to be ready for prime time and broadcast by early in the fall. Just to show you what the design looked like, the patients were randomized 1-to-1 to a 3 times daily exposure to oral Ubenemex versus placebo. We had a primary endpoint of looking at skin thickness thickness in the skin, with secondary endpoints of histology, limb volume, bioimpedance, and patient-recorded outcomes, and the efficacy evaluation was after 6 months of exposure to the drug. I'm going to briefly talk about one more project, and then we're all going to go home and be happy, I hope. And that is to talk a little bit more on the research side related to surgery. We've heard a lot today about LVA and specifically about vascularized lymph node transfer, and I've been interested in the latter issue. We know it's highly successful, and Dr. Dayan showed us a lot of the evidence for why that is so, but we know it's not universally effective. And what we know from experimental animal models is that when vascularized lymph node transfer doesn't work, it seems to be because of failure of engraftment of the lymph node into the existing and surrounding lymphatic circulation. And that happens and has to happen in a time-dependent fashion, and it isn't always able to do so. In order to address that issue, I've worked with a startup company in the Bay Area to develop this product, which we call the BioBridge. It's basically a biological scaffold that, to the naked eye, looks like a piece of suture, but it's composed of collagen, normal mammalian collagen. The telopeptide is cleaved from the molecule because that's antigenic, so we remove that, and then through nanoprocessing we highly align the fibrils so that they look like the image that you see here, and in cross-section you can see that this is a very porous structure with highly aligned longitudinal spaces. And basically, to make a very long story short, lymphatic endothelial cells love to grow across these clefts, and they very quickly will line the scaffold and grow across it. And when we look at lymphatic endothelial cells, these are human lymphatic endothelial cells in tissue culture, they align themselves in a very physiologic, longitudinal, overlapping fashion that suggests that they are healthy and that they are growing in a normal fashion. And so this gave us great promise for the notion that if we placed these scaffolds in apposition to our surgical site, perhaps they would accelerate the engraftment by basically serving as a bridge between the lymph node that's being transplanted and the surrounding matrix of vasculature to which that lymph node has to connect. So we looked at this in a large animal model in the pig, and the pig gives us the benefit of having a single draining lymph node in the limb, so that lymph node could be surgically extirpated, and then we created a field of radiation therapy that you see pretty easily in this figure that would attempt to simulate as much as possible the post-cancer treatment effect that would lead to lymphedema. And again, to make a long story very short, what we found is that when we harvested the tissues at the end of a 3-month period of observation, we were able to see new vessel formation across the BioBridge. These had the immunohistochemical characteristics of new lymphatic collecting vasculature, and you can see the remaining collagen fibers that are in close proximity to these newly formed vessels. I can tell you that all of this collagen is both biologically inert and is resorbed by the body in a period of 6 to 9 months. It's gone after it does its job. So this is the immunohistochemical proof that these are new lymphatic collectors, and correlated with that finding, we saw resolution of the lymphedema both by imaging characteristics as well as by tracking with bioimpedance. So based on that information, the FDA has granted me an investigational device exemption to test the BioBridge in human patients, and our project is funded by the National Cancer Institute. We have a 3-year grant to look at the comparison between vascularized lymph node transfer as it is currently performed unaided versus lymph node transfer using the BioBridge as an adjunct. We did treat our first patient in June of 2017, and that patient has actually completed the study by this point. The study is still actively enrolling. We are going to extend the study ultimately probably to a total of 4 sites where the patients will be enrolled for a total of 60. It will be a 4-to-1 randomization between exposure to the BioBridge versus the standard surgery. The basic concept is we place 5 to 10 of these BioBridge scaffolds both proximally and distally to the vascularized transfer, and there is just a light suturing of the scaffolds to the surrounding soft tissue. It's not a direct anastomosis with any vascular structures. We simply then rely on biological forces to bring the lymphatic endothelial cells across the scaffold and toward the BioBridge, toward the transfer. So this is a study of adult patients up to 75 years. The FDA has asked that we limit this study to breast cancer survivors, so we'll be looking only at upper extremity in the study, but the notion is that this would be similarly applicable ultimately to lower extremity VLNT. They have to be 3 years out from their cancer therapy and free of clinical disease, because the FDA wants us also to demonstrate conclusively that there is no risk of cancer recurrence associated with this intervention. They need to have involvement of one limb only, and of course they have to be eligible for the primary surgical intervention. They have to have completed decongestive therapy and be using compression garments for at least 8 weeks without any change in the regimen up until the time of surgery. So this was our first completed patient. She did receive the BioBridge, and you can see that her right upper extremity, which was previously the lymphadematous one, is nearly normal. She also had a tremendous problem with breast lymphedema, which is pretty much completely resolved since the use of the BioBridge. Obviously, I have no comparators to show you, and this is not intended to be a conclusion of the study, but simply to say we're very excited to learn what this can do. Of most excitement to me, and this is more late-breaking news, is that we've gone back to more of a small animal study in the rat, actually, and have created surgical lymphedema in the rat hind limb, again, by extirpation of lymph nodes and radiation therapy. And in this case, we are using the BioBridge in more of a preventive approach. So at the time of surgical intervention, we're simply placing the BioBridges across the site of lymphatic extirpation. And basically, what we were able to find is that when we place these BioBridges, we are encouraging anastomosis to the contralateral limb, and basically there is no lymphedema that develops when we use the BioBridge at the time of the surgical damage, whereas in the control subjects there was a significant lymphadematous development and very little collateralization to the other side. So we're hoping, so this is what one of the treated animals look like, and you can see the clear migration by fluorescent imaging from the ipsilateral limb to the contralateral side. So implantation of the BioBridge at the time of lymph node excision prevented development of lymphedema. It encouraged development of a new lymphatic vasculature with collaterals crossing the midline toward the contralateral normal vasculature. So all of this, I think, lends great hope, not only these studies, but things that people are doing all around the world for all of those new publications that are appearing in exponential quantities in the literature. And our hope is, the sky's the limit. Maybe one day lymphedema will really have a cure, and that is my dream, and I think we should all participate actively in trying to make that happen. So thanks very much for your attention just now. for your great attention all day for this long day full of facts and information, and I hope this has been useful to everybody who's been here. Thanks very much. I just received a question about Ubenimex, which I understand might be high on everyone's list of curiosity. So let me explain a little bit about where we are with regard to Ubenimex, because there's been a headline recently in the news. Many of you probably know that the outgrowth of my research over really the last 10 years has been this clinical trial of the drug that we discovered in response to that research, and that trial was called ULTRA, to look at Ubenimex. It was an international study. It was conducted in 4 sites ultimately, 3 in the United States, 1 in Australia. The company that sponsored this study defined a very narrow primary endpoint, and sorry, I've been asking to turn up the audio. That's pretty much as high as it goes. I'm sorry. I've got a strong signal. It's there. So to continue, I'm sorry, we had a little technical issue there. To continue about Ubenimex, the primary endpoint as defined by the sponsor was not met, and therefore they decided to discontinue the program. That's the headline you may have heard about. I will tell you that I have had a first top-line look at the data, and what I see very encouragingly is a responder population. We feel that this is worthy of more study. Much of the data that was collected in the trial has not actually been looked at yet, but we will be doing this over the weeks and months to come. The project is not dead, and I still believe very much in Ubenimex and LTB4 antagonism as a way to treat lymphedema. I certainly can tell you that in the group of patients that I treated at Stanford, there were individuals who basically had complete remission of lymphedema while under treatment, and so we want to continue to develop this for future use. So somebody said, I missed the dosage, and I assume this is about ketoprofen. The dose that we used in the trial was 150 milligrams 3 times a day, but that preparation, apparently, at least in the United States, is no longer available by the generic manufacturers. So more recently, I've been using 225 milligrams of the long-acting preparation once daily. The next question is, would it be safe to self-trial? The answer is no. This definitely must be accomplished under the guidance of a healthcare professional. First of all, at least in the United States, ketoprofen is a prescription-only drug, and I have to say that one of the big reasons for trying to develop Ubenimex is that there are safety issues with non-steroidal anti-inflammatories, including ketoprofen, and a risk-benefit analysis would certainly need to be undertaken by a healthcare professional who would be able to guide the patient. Now, the next question is the trial about ULTRA is completed. There is no more enrollment, but it did include both primary and secondary lymphedema. I will be continuing to look at Ubenimex, at least here at Stanford, and again we will be looking both at primary and secondary lymphedema. Now, I have a question about advancement in the surgical intervention of breast cancer and how surgeons are preventing lymphedema before it occurs. Fundamentally, there are 2 strategies that we can talk about in terms of prevention, which is different than the treatment program for lymphedema. Sorry, my camera just shifted.
The first is what's called axillary mapping, reverse mapping, and this involves a variation on what currently is accomplished in sentinel node identification. Some of you are probably aware that in breast cancer, the surgeon will inject a substance into the breast to look for its drainage into the first draining lymph node that is encountered as the lymph leaves the breast. This is called the sentinel node. One can do the same thing for the arm by injecting a different material concurrently into the arm and identifying the sentinel node for the arm. In theory, if one avoided the sentinel node for the arm and still could identify the breast sentinel and remove it for analysis, one could significantly limit the amount of lymphedema that one would encounter. Now, sometimes the breast sentinel and the arm sentinel are the same, in which case there is no option. One has to go ahead and remove it. But when it is possible to save the arm sentinel, that would reduce the risk of lymphedema. A second approach is called lympha, which basically involves a more complicated approach at the time of breast cancer surgery when the lymph nodes are removed to create a lymphovenous bypass at the time of the surgery in order to try to prevent lymphedema, and there are very early trials to suggest that this might be helpful. Somebody asked about artificial lymph nodes. It's something I'm very interested in, although nothing is available to date. It's really only a theoretical concept, but I do believe that if one could create an appropriate scaffold, basically one could populate this scaffold with the appropriate lymph nodes and then place that lymph node or lymph nodes into the axilla to replace the ones that are removed. This is a very futuristic idea of medicine that probably is at least 5 to 10 years away. Somebody has asked whether we will be doing any trials with Ubenimex for arm lymphedema. Yes, we will be looking at arm lymphedema here at Stanford now that the ULTRA trial is completed. The decision to look at legs only was one that was created by the sponsor at the time of the creation of ULTRA. Now that ULTRA is done, we're going to be looking more broadly at other topics as well, including arm lymphedema, including primary lymphedema. We really want to understand, how does this drug work, what is its place, and can we develop it as an effective therapeutic, in my estimation, for hopefully all forms of lymphedema. Let's see, I don't know that we have any other questions at the moment. Let's see. So somebody asked, did the ketoprofen people get limb reduction as well as improvement in skin? Yes, not everyone, but I do want to hasten to say one of the limitations in reducing limb volume, at least in a short-term trial with ketoprofen or Ubenimex or any drug that we might choose, is that by the time somebody has had lymphedema for a period of months to years, the majority of the limb enlargement is fat overgrowth. And fat overgrowth, if it is going to respond to the strategies that repair the lymphatics, will take a much longer period of time than a 4 or 6 month trial in which we're looking at these drugs. So that represents a next frontier for investigation, but we do believe that these drugs work by allowing the body to repair the damaged lymphatic system, and therefore they do result in some improvement in the skin biology, which will directly influence the rate of infection and the functional ability of the involved limb to be as normal as possible. But size is going to reflect a lot of the fat overgrowth that's present, which, if it's reversible, will take a longer period of time. Herantis lymphactin is a question. No, at the moment, lymphactin will not be studied here at Stanford. At least we've not been invited to do so. Those studies are going on primarily at centers in Finland, and it is an attempt to try to make lymph node transfer be a more effective surgical therapy. I'm not involved in that particular work, although I was in the early stages of it. I'm not currently in discussion with those investigators. Ready for a few more questions if there are any. Well, it looks like I have answered what's most on your mind. I just want to reiterate that there is a big paradigm shift in the treatment of lymphedema. For my entire career, this has been considered an irrevocable disease that can be managed through physical means but cannot really be addressed by standard pharmacologic interventions, taking drugs to treat it, as we do most chronic diseases in adults and many children as well. Now we have a class of drugs that I think does work, and the biology is very exciting. I think we can change the face of lymphedema. We just have more work to do to understand how best to do it. Somebody is asking, or a couple of you are asking, how we are enrolling additional studies and additional patients in these trials. At the moment, this is going to be work only at Stanford, so anybody who is within shooting distance of this institution can certainly inquire through LE&RN, through any contacts that you have for me or my coordinators. But unfortunately, if you live at a distance from Stanford, we won't be able to help you. So I think that maybe will take us to the end of this set of questions and answers. Please don't hesitate to send any questions you have in writing by email if things come up in the future. And thanks so much for participating in this webcast. Thanks so much, everyone. Bye-bye.